Okay, everyone. Welcome to January's Scholarly Snippets session. This is a 30-minute, really quick walk through a topic near and dear to me, how to read a scientific paper. I'm Kevin Bradford, the Research and Writing Support Librarian at Philadelphia College of Medicine, their Georgia campus. Metro Atlanta is where I'm located. And we're going to go right ahead and let's see if I can get that out of the way. That little pesky bar, that down at the bottom, and minimize the people screens. So, okay, hopefully this makes it a little less cluttered. Okay, so what we're going to talk about is how to read a scientific paper. And typically it's formulated in IMRAD format, the basics of it, the introduction section, the materials and methods, the results section, and the discussion and conclusion section. There are, of course, other parts of it, your title and author, your abstract or your summary, and then your acknowledgments, references, figures, and tables. But for this presentation, we're going to pretty much focus upon the introduction, materials and methods, the results, and the discussion/conclusion. Now, sometime along in the 1990s, the scientific community, publishing community, decided to get together and try to make published articles a little bit easier to read in terms of their format, because they were finding that the different sections of a scientific paper were kind of like helter-skelter all over the place, depending upon the journal and or the actual article. Some journals are a little bit more loosey-goosey on this format now than most. Pretty much most academic journals want your galley proof from the writer or the drafts to be in the IMRAD format, but there's a lot of breathing room within that as well. Some papers separate out the materials and methods. Some people will put the discussion section right in with the results, have the conclusion section, separate, or they might include it in a discussion conclusion section together. So, but typically it'll follow that flow. There'll be the intro, materials and methods, results, discussion, and conclusion, typically. All right, now as you're looking at your paper, bear in mind, of course, we all know how to read, or we wouldn't be where we're at. So this is not about how to read. It's about how to use your time more efficiently when you're parsing scientific articles, because by their very nature, they're going to be very dense pieces of writing. So you can tell as you've got your articles, you've done your research, and you're taking a look at your articles. I'm kind of setting this up in your mind. You've got a stack of studies, and you're going to go through these to see which ones are relevant, which ones you can toss. So first thing is to take a look at the title and the authors. The title is usually pretty descriptive of what the topic is. It'll often state within the title the author's main finding. And the order of authors is also important. What can you tell from the example that's down below? The article is VEGF, pro-survival factor, which acts in concert with TGF-beta1 to induce endothelial cell apoptosis. And the order of authors indicates here the first author is the individual who is primarily responsible for the creation and publication of the paper. It may not necessarily be the experimenter in a particular scientific experiment. Usually it is, so it's usually the chief or the lead, and they will have responsibility for the majority of the bulk of the paper. So if you are aware of authors within your field and you know who they are, this might be a clue for you that, okay, well, this is Dr. Ferrari's known expert in X. And so this article I probably want to take a look at because he or she is the acknowledged expert. So that's one little tip for you. Okay, so the abstract and summary is typically what you see when you do, say, a PubMed search, an Embase search, a search in any of the subscribed databases that medical libraries and other libraries will subscribe to. And so that's a labor saver. That's the first place really you want to take a look at once you've parsed the title, once you've looked at the title. Go right to the abstract or the summary. if one exists. Most scientific index papers do have abstracts available. So this is typically what you would see when you do a PubMed search. You can often decide if the paper is worth reading based upon this information. There'll be a brief background of the subject matter in the paper. The abstract will contain the purpose for the study. It'll also give you a very brief blurb on the major findings and also the relationship between the findings of that particular study and the field that it's related to in general. Now we're going to move quickly on to the introduction section. And oh, by the way, I'll open it up at the end of the session, at the end of my lecture for some Q&A if you've got any questions, burning questions at the end. So if you'd save those for the end, that'd be great. Okay, so the introduction section in a scientific paper typically presents the background information for the community or fellow scientists. Typically, these papers are written for a scientific community, a specific scientific community, and also for knowledgeable persons in other similar related fields. And the idea is to convince and to show and to demonstrate that the particular experiments that were carried out have a value and a worth for the scientific community to address and take a look at and study. So the structure and introduction is usually, again, I say usually because sometimes you'll find variances. It'll start out with the accepted state of knowledge in the field if it's a question on a therapeutic approach for a particular patient. The paper will address what's currently being done. Or if nothing's been done, it'll indicate that as well. But typically your gold standard treatments, those will be mentioned. And then the focus will move to a particular aspect. Often it's the sets of data that led directly to the work of the particular paper that you're reading. So also going to include the hypothesis being tested. Most journals are very, very particular on this point. Your hypothesis needs to move in quickly on your paper. Usually the introduction, if it's not indicated by the end of the first paragraph, if the introduction is a little longer, say 2-3 paragraphs or so, your hypothesis needs to move in quickly. So when you look for that, if the paper is formatted properly in IMRAD, that should be toward the end of the paragraph, the introductory section, however long it might be. There's where you will typically look for it and find it. And then there'll be brief conclusions in the introductions. Scientists and members of scientific communities, none of us like surprise endings. We don't like to weed through the woods, to get the answers to our questions. Give us the answer that you found and give it to us quick and fast. So typically the conclusions will also be in the introduction, as well as a hint, as I'd mentioned earlier, perhaps even in the title of the article. So how to approach the introduction? I am pretty much a Microsoft Word person. I tend not to do a lot of drafting on paper anymore. I'm just kind of so honed, if you will, for an electronic sort of a blank rather than a paper blank. But if you are a paper person, this is typically what you would do. Grab that piece of paper and start your notes, draw those many figures, anything that will help you if you're a visual learner, then certainly, your many figures and graphs and charts on your own will be helpful. Define the vocabulary. You can highlight if you don't understand certain terms. And it's certainly okay to use Wikipedia or a medical encyclopedia to look up those terms. So the questions you want answered as quickly as possible in your parsing is what is the accepted state of current knowledge in the field? What data led directly to the work of this paper? You want to look for the hypothesis, and you want to find the conclusions. And again, that's usually stated in the last intro paragraph or in paragraph 1, 2, or whatever. So that's what you're needing to address on your blank piece of paper. Typically, what I suggest is leave a lot of white space in your categories in your notes. So that way you can go back and fill things in rather than having to scratch it all out and start over again. So leave a lot of white space for you to do a lot of edits as you're taking notes. Now, in the materials and methods section, these should be detailed enough for another scientist to be able to replicate the work. The volumes that are used in the experiment, the number of times a particular experiment is tried, the control groups that were used, who gets the placebo, who gets the gold standard, who gets the new treatment, et cetera, so forth. How many participants were there in each group? Were they fully blinded? Was it partial blinding? What are the materials or patented therapeutics? Now here, it's not in this particular workshop, but I'm in the process of drafting a new PowerPoint slide for this. So in the future, when I present this, it'll be included in there. But some of our faculty here have pointed out that they are, unlike me, they are visual learners. They are visually tuned, whereas I'm tuned to text pretty much. That's been my background. So what one faculty recommended that he does, and it doesn't necessarily turn all this, what I'm talking about, on its end, but just consider it if it works better for you, is to do the quick and dirty on a paper. Go to the conclusions. He'll typically look to see what the conclusions are. He'll identify if it's the topic he's looking for. He'll look for the conclusions. Then he will look at all the graphs and charts, and he will read them and see if they're copacetic with the conclusions in the paper. So that's really actually in a couple of minutes, that's the quick and dirty way to parse and read a scientific paper if you're visually oriented. But of course, you can't make a complete decision as to the value and worth of the study or the paper without going through all the rest of the material that I'm covering here. So, but it could make a decision much faster for you if you're visually oriented and you're comfortable with graphs and charts to do it that way. Grab the conclusion, read the conclusion, go to the graphs and charts and see if they match up. So going forward in the materials and methods section, in reality, this is often compressed in text. And because of that, you might need to look up another paper that is referenced for more detail on the process, because simply science builds upon previous science. And so there may be materials and methods that are commonly used. Then in this particular case, the particular paper you're looking at might be true, but the new paper uses those common materials and methods in a different sort of a way. So there's not going to be a lot of background in terms of how they put it all together physically and logically. So if there's not enough information in the materials and methods, probably you can assume that this is a process. Most of the ingredients, if you will, of the experiment have already been gone over, and most researchers would be fairly familiar with that. So I won't kill that any longer. We'll move right along. So the question of it is should you even read the materials and methods section of the first parsing? Well, yes and no. It's what you're comfortable with. Often you can skim over the materials and methods and go right to the results or the conclusions, like some of our faculty here have told me that's how they do it. Pick out the conclusion, go right to the graphs and charts, go right to the text part of the conclusion. However, when you get to these results, you'll need to refer to the methods to clarify how the experiment was actually done. So you can get those sample numbers, what are the conditions and specifics of the study that you need to take a look at. So basically reading a scientific paper is a stepwise process. The first, what I'm talking about here, is you're going in and you're zooming through it. Basically, you're giving a quick parsing in all these sections, then filling out your notes, then you're going to go back in for clarity. You'll return for clarity. And in that process, you'll be able to weed out a lot of studies and papers that aren't relevant. Okay, so the results section. While the introduction section typically poses the questions that are going to be asked in the paper, the results section usually describes the outcome of the experiments that were done to answer the questions. So our results are often simply stated, with the interpretation of them coming later in the discussion. So you will find perhaps any graphics may wind up in the results section. They could also wind up in the discussion section. They could be scattered throughout. your images, your visuals may also be saved for an appendix in the article. They might be saved for a separate section at the end of the paper. That's not typically what I find in most papers. Are scattered throughout the methods and the results, et cetera. So, and again, figures and tables allow you to see the outcomes of the experiments for yourself. Okay, so how to read the results. My first recommendation is read the text straight through, but as each figure or graph or gel is referred to, examine it and then look up what you are not familiar with. Here, take your notes, giving yourself a place to refer to about each figure, so plenty of white space. So as you're going through and you're looking at these visuals, comparing them with the text, the little yellow box down here, there's 5 questions you should be able to explain. Should be able to explain it to a fellow classmate, fellow colleague. These 5 things are: you need to be able to talk about the basic procedure in your own words, the question it sought to answer, what the results were in your own words, what the conclusion is, and note any criticisms. These can be from the experimenters themselves, where they're taking a look at their study and they're saying, well, there's certain aspects of our study that, maybe we didn't have enough persons allocated to certain groups. Maybe some volunteers in the groups dropped out of the study for whatever reasons, and so we had to reallocate. So these are all things that you want to take a look at, because anything that's done to modify the experiment once it's underway, I should say, is going to be a valid criticism in terms of being replicable to, or generalizable, if you will, to a particular patient group, particularly a single patient, your patient. So you should be able to explain these things, any kind of variances and differences, and look for what the authors have found. Also, in terms of criticism, your own criticism as well. Sometimes the authors don't pick these things out. That's why we have a community of scientists, a community of experts for all feedback purposes. Now, in the discussion section, sometimes this is mixed in with the results. Sometimes the discussion is followed immediately by the conclusion. Sometimes they're all separate subparagraphs in a paper, but they generally follow this flow, as I'd mentioned, that we're showing here. In the discussion sections, typically the data is analyzed by the authors to show what they believe it actually demonstrates. Now, you don't have to necessarily agree with the interpretations of any individual paper. Your task in reading it through is to just note what the data is and what the authors or experimenters are saying it means. You're also looking for a discussion about how their findings are related to other discoveries within the field. How does their particular study or paper contribute to known knowledge? How does it correct errors, perhaps in a therapeutic approach? And basically, you're looking for the authors to tell you why is this paper worth reading? Why is this experiment significant? What does it have to contribute? So all spaces for your notes fields. Okay, so how to actually dig into a discussion, take notes. Again, what conclusions do the authors draw? Be sure to separate fact from their opinion. That should be fairly easy for you to do. If you have any questions regarding what opinion and fact are, certainly you can reach out to me and I can help you with that. A lot of times that is obfuscated. So you need to be able to describe for yourself why these data are significant, as I just mentioned. And, well, that's basically it. I moved through this really quickly. Again, this is designed for a workshop, and our scholarly snippets are more in a lecture type of a format, so we missed that end of it. In the real world classroom, I would be tearing a paper apart. So, but hopefully this is logically paced enough so that you'll be able to take that paper, pull up this recording, and be able to follow it through that way. So in the references, you'll find papers cited within the text. You'll be able to discern also what parts of the paper cite other papers, and that may lead you on the breadcrumb trail to other studies and papers that you might not have found when you've done your literature search in the databases. definitely something to take a look at. What parts of the paper cite other papers? Typically, the introduction doesn't cite much unless it's citing something very specific. You typically won't find citations in an introduction. You'll find quite a few in materials and methods. You'll find a lot of them in the discussion section, and there might be a few citations in the results section. Where you should never find citations is in the abstract or the summary section of the paper. That should all be in the author's own words. Okay, here I have another method that I base this talk on. Anne McNeil has a 4-step method, and I have a URL link there as well that you can refer to in the recording when you pull up the recording. So, and again, her answer is not necessarily in order, and that would be mine as well. So I'm just going to skip through this because it basically repeats what I've already said. So, I mean, you've done a lot of work, but you need to save your energy for overall reflection and criticism of the paper now that you've read it through and you have all of your notes. So this last graph slide, or next to the last, yes, next to the last, is what I do in active learning workshop sessions where we're pulling apart an actual paper. These 6 bullet points are crucial and very important for you to be able to answer, for your students to be able to answer. Do you agree with the author's rationale for setting up the experiments the way they did? Why or why not? That needs to go in your notes. Did the authors perform the experiments appropriately? Did they repeat them adequately? Did they use the correct control groups? Did they have enough participants to mitigate for any kind of randomization of results? Did they use appropriate measurements? Were there enough experiments to support the major findings the authors are claiming in the paper? Do you see patterns and trends in their data that could be problems that they missed? I alluded to that a couple minutes back. Do you agree with the author's conclusions from these data? Are they overgeneralized or are they too grand? We're not looking for beautiful science. That's one of the tip-offs if you find a paper that says, we have found the answer, and the answer is only within this particular experiment. Usually they won't state it out in such a bold way, but if that's the end result, that certainly is a red flag, because science, no one experiment is the be-all and the end-all of knowledge on any particular topic. Science is continually growing. Science is continually refining itself. Science is continually discovering new processes. So then the last point would be, what further questions do you have? What might you suggest the experimenters do next? And of course, this is for you. If you need to know what you think that they should do next, or what would make this a better experiment or a more convincing experiment. So that is it. It was a very, very quick run through. And now, if you've got any questions, I would be more than happy to address those. I'm going to stop sharing here. And here I am back in the room. I hope this was helpful. Everyone's on mute. You can unmute. Don't be shy. We've got about 5 minutes. Oh, yes. Let's see if I can put that in chat. Chat. Okay. So the link for the methodologies was requested. Let me see, where was that? Here it is. And this is the Anne McNeil method. That should do it. Okay, there it is. There's the link. And let's see. Another one asked for the link. And I think that's it in text. Okay, so all caught up. Thank you very much for attending. I hope this was helpful. And I'll give you my email just in closing.